All right, guys, so welcome to the BRI Short Talks. These are new assistant professors that have joined the BRI. We're really excited to hear about your work. I'm going to be hosting on behalf of Tom and Felix today, and we just want to thank everyone in the BRI for a really successful year, and it's exciting to be back sort of in person to be communicating about all our science again. It's been a really fun year. We also want to give a really big thanks at the end of the year to our BRI office, who are amazing. They really are just like a squad team. You just kind of know that everything's going to be taken care of. So we're really grateful to them for basically running the show. And we want to wish everyone a really happy summer. We have 4 short talks today from our junior members of BRI, and the first one is going to be Amjad Askary. He's an assistant professor in the Department of Molecular Cell and Developmental Biology in the College of Life Sciences, and he completed his Ph.D. in genetics at USC in 2016. He then went on to Caltech until 2021, so he's managed to stay in Los Angeles for his whole career, which is really the dream, right? He's had a number of awards, most recently a K99, and also a very prestigious HHMI Jane Coffin Child's Postdoctoral Fellowship Award. And today he's going to be talking about lineage and cell fate determination in the mammalian retina. So we've got 10 minutes to talk and 5 for questions. So take it away, Amjad. Amjad, that was really interesting. I thought it was really interesting to think about how it might be related to other parts of the brain and the genetic determinants of different cell types in different regions. Did anyone have any questions? Okay, I'll ask one. How do you think it might be related to different regions and different cell types and determine particular things in the rest of the brain?
Yeah, so I should say first thing, so far what we're looking at are the lineage trees and the lineage relationship.
So while they're setting up, I'll just introduce Bridget. So Bridget Callaghan is an assistant professor in the Department of Psychology, which is also where I am. This is someone I've known for a long time, about 15 years. We actually did our PhDs in the same institute. So it's really cool that we've ended up at UCLA in the same department together. She's someone I've always looked up to. So she completed a PhD in Australia, as I said. She moved to the US to complete a postdoc in Columbia, and then moved here to UCLA in 2019, just before COVID hit. She has many awards as well. Most recently, the Federation of the Association of Behavioral and Brain Science Early Career Impact Award in 2021, and also the very prestigious Association for Psychological Science Rising Star Award a few years back. And she's going to talk about the generational impacts of early adversity on children's mental and physical health. Take it away, Bridget.
Okay, thanks, Mel. That's a very, very nice introduction. We have known each other a long time, so it's really nice to be here at UCLA. We were having a chat before we got started, so I've actually been here at UCLA since the end of 2019, but it's really nice to kind of be back in person and to be joining this community. So I'm really looking forward to future interactions with you all. So today I'm going to look at the generational impacts of early life adversity on children's mental and physical health. So this might be a bit of a jarring change from what Amjad was talking about. So we're going from cells all the way up to humans. So it's going to be a little different. And particularly I'm looking at the generational impacts of adversity through these pathways of brain-gut microbiome access. So I'm cheating a little bit. This talk does have some brain in it, but it's also got some other non-brain systems as well. So in my lab, we're really interested in looking at early life experiences, and particularly early life experiences with the caregiver. So we tend to study things like maltreatment, abuse, neglect in childhood, and how these influence an individual's emotional as well as their physical health. And in doing so, we tend to look at a range of different biological systems that we know underlie an individual's emotional and physical health. So in my lab, we tend to focus a lot on the brain in our head and the one in our gut. So that is the gastrointestinal system. And we also look at an organ that a lot of us don't think about that much, or at least historically we haven't thought about that much. And that is the microbes that live inside of our gut, inside of our gastrointestinal system, and that participate with the brain and gut in this bidirectional communication highway called the brain-gut microbiome axis. So in my lab, I look at the influences of early life adversity on each of these nodes of the brain-gut microbiome axis. How do you make this work? Oh, no, anyway, whatever. So on each of these nodes of the brain-gut microbiome axis, and then I look at how each of these nodes influences an individual across their lifespan, even into when they reach adulthood, potentially affecting the next generation and possibly even multiple future generations. So that's really the model that my lab is working under at the moment. But right now, today, I'm going to just tell you about some of the most recent work we've been getting up to. So that's focusing up the top here, looking at the impacts of adversity across generations on brain as well as the microbiome, and then tell you a little bit about how we're trying to look at the interactions between the brain and gut microbes. So I'm going to show you two studies today. Both of them are taken from this really great community cohort called Growing Up in Singapore Towards Healthy Outcomes, or GUSTO. And we have been lucky enough to, right as COVID started and my lab had a lot of trouble collecting data, we were able to pair up with Michael Meany, who's a researcher who's been really integral to this study, and be able to access some of this data. So we've been really lucky, and I want to say an enormous thank you to them. Okay, so I wanted to also highlight the work of some of my students. So this student here, Dr. Jessica Uy, she actually did her PhD here in the lab of Adriana Galvan, another BRI member, and she's now a postdoc in my lab. And she was using this GUSTO cohort to look at the generational impacts of adversity on brain and behavior. So in this cohort, she had 541 individuals. We had mothers who were retrospectively reporting on their adverse childhood experiences, so issues like maltreatment, child abuse, neglect, and physical neglect as well. And then we had those same mothers reporting on their own anxiety and depression symptoms when they were pregnant, on their postnatal anxiety and depression symptoms. And then we had children's parent proxy reported and child self-reported emotional health when the child was 7 years of age. So these are second generation children. So what Jess found in this study is that maternal childhood maltreatment was directly associated with parent proxy reports of children's worse emotional health, so more maltreatment, worse emotional health in the child. And this occurred indirectly, serially, through a pathway of worsened prenatal as well as postnatal mental health. She also found an association for children's self-reported emotional health, and that was operating through a pathway of worsened, specifically prenatal mental health. So maltreatment, worse prenatal mental health, and then worse children's self-reported emotional health outcomes at 7 years of age. So in a much smaller subsample that had good quality resting state MRI data at 6 years of age, so that's very difficult to get. So we have a much smaller sample size here of 89 6-year-olds. We found that mothers who were reporting high levels of anxiety and depression during the postnatal period, over and above maternal childhood maltreatment and maternal prenatal anxiety, that was associated with second generation children's amygdala prefrontal cortex resting state connectivity. And that this resting state connectivity pattern was associated with 7-year-old children's subsequent emotional health. So that was both parent reported and child self-reported. So clearly, Jess in this study here is seeing some evidence for these intergenerational impacts of early life adversity that really do seem to be operating through the pathways of maternal, prenatal, and postnatal mental health, impacting children's brain as well as emotional behavior in second generation. Now, a graduate student of mine, Frank Hadassi, she has been using this same cohort, so very same kind of setup, but she's actually been looking at children's microbiome at 2 years of age, so second generation children's microbiome. Only difference here is instead of looking at maternal anxiety and depression, it's a composite score of anxiety and depression, as well as any adverse events that have occurred from the child from the time of birth until 2 years of age when we collected the microbiome sample. And depending on, what adversity measure was included here, we have a sample of 285 to 450, which is pretty big for a microbiome study. So that's a neat thing about this. So Fran finds different things depending on what type of adversity we're looking at. So in terms of alpha diversity, so this is a within individual metric that basically looks at how rich and evenly distributed the bugs in a person's gut are. She sees that only postnatal adversity over and above the other forms of adversity were associated with lower levels of alpha diversity in 2-year-old children. Beta diversity is a between individuals metric. So this is looking at a distance matrix of how similar the bugs in my gut are to everyone else's. And we find no differences in beta diversity across these different types of adversities. And then finally, with differential abundance, we find unique effects of each type of adversity on the differentially abundant bugs. So specifically for maternal childhood adversity, Fran found that it was positively associated with this bug, Clostridium sensu stricto. This was an interesting finding because this bug is typically associated with butyrate production, which is something that we typically think of as being positive. However, it's not a very efficient butyrate producer, and it's usually outcompeted by more effective butyrate producers in the gut. So we're thinking that this may actually be an ineffective, a marker of ineffective butyrate production in the guts of these individuals. For prenatal adversity, we see lower rates of lacnoclostridium. So this bacterium is an efficient butyrate producer. So here we're also seeing a picture of less efficient butyrate production in the guts now of children whose parents have been exposed to prenatal adversity. And then finally, postnatal adversity was associated with lower levels of Parabacteroides, generally associated with health, and high levels of Finegoldia, generally associated with inflammatory states in the literature. So again, some sensical associations there. So what have I shown you today? I have shown you that second generation children of mothers who've experienced adverse childhood experiences, it impacts the second generation's children's emotional health. It also impacts their brain in terms of amygdala, prefrontal cortex, resting state connectivity. And it seems to impact their gut in terms of these different bugs and alpha diversity. And it seems that many of these pathways, at least the emotional health and the brain, are operating through impacts of early life experiences of adversity on maternal feelings of anxiety and depression, both in the prenatal and in the postnatal period. So what I haven't shown you today are some work that's ongoing in the lab where we're trying to link these brain patterns in the Gusto cohort with some of the microbiome as well as the emotional health outcomes. And I also haven't shown you another huge side of our lab, which is to look at exposed individuals, so generation 1, impacts on their physical and mental health, microbiome, gastrointestinal symptoms, and brain function. But that's a good chunk of what we do in the lab as well. So outstanding questions that I wanted to highlight, so other things that we're doing at the moment. So we need to know a lot more about women who've reached childbearing age, who have experienced adverse childhood experiences. What is the impact, ongoing impacts of those experiences on their brain-gut microbiome axis? How are these experiences impacting their physical and mental health, and how do those things interact? We have a study that came out pretty recently looking at cognitive plasticity during the period of pregnancy in women. So this kind of goes against this idea of baby brain. So we found some high cognitive plasticity in pregnant women. So we're now looking at the neural correlates of that, as well as seeing whether that is impacted by a mother's experience of adversity. We also need to know how long these effects last. So in the second generation children, we haven't really looked beyond 6 or 7 years of age. So how long are those lasting? And we also need to know about moderation impacts. And so in particular, in my lab, we're really interested in the parent-child relationship and how positive parent-child relationships can kind of overcome experiences of adversity. And so another postdoc in my lab, Dr. Jen Summers, as well as Dr. Wee and Fran Kadasi, my graduate student, have some work on this during COVID and continue to look at it, and we'll be looking at it in the context of microbiome. And finally, we need to know a bit more about different other microbial niches. So a current graduate student of mine, Naomi Gantz, is looking at the oral microbiome and how that is impacted by early life adversity, as well as looking at development specific factors. So this is an indication of puberty here. So how does puberty influence the microbiome and subsequently plasticity within the brain that we know is really high during pubertal stages of development. So I have a study in my lab that's going on at the moment, just to tell you about it, because we're analyzing some of the first wave of data now. So it's a staggered longitudinal design. And by the end of each of these 3 waves of the study, we would have covered each of the ages of 6 to 11 and then 13 to 18 years of age. We're looking at a group of comparison kids who've always been with their biological birth or first families, and then a group of children who've experienced really extreme caregiving adversity in the form of foster care here in the United States, followed by adoption. Wave 1 is complete. We're doing metagenomic sequencing on the microbiome data. So the past studies I showed you were taxonomic. We could only say what bacteria were in the gut, and here we'll be able to say more about what they do. And we're doing some neuroimaging in wave 2. So that is starting up right now. So we'll be able to look at some of those functional profiles in the gut and how those associate with the brain. So I wanted to say a huge thank you to my wonderful lab, most of them who've just been around during COVID. So we've only really had a lot of online interactions, but they also do really amazing things like travel out to the desert right before Christmas to try and recruit families into our studies. So they really go above and beyond. They're fantastic students to all of the families and youth, and to our funding sources, and to you all. I'm happy to take any questions.
That was great. Thanks, Bridget. We had a question up the back. People on Zoom, also feel free to ask questions in the chat.
Hi, Bridget. Really interesting talk, and it's a little depressing. I don't mean this as a criticism, but it comes off a little fatalistic. That as a mother, if you had a difficult childhood, you know, the implication is that you're then condemning your children to a lifetime of anxiety and depression. Have you considered looking at mechanisms of resilience, and related to that, do you see the microbiome as potentially an interventional target, a therapeutic target that could be used to protect against these intergenerational effects?
Yes, I wish that I had included some of my rat data that really spurred this line of research in humans. So in rats, we see the intergenerational transmission of maternal separation impacts on fear learning early in life. So we see that in 2nd and even 3rd generation pups who had a father who was exposed to maternal separation. And we actually found in several different studies that were subsequently independently replicated by a group in Taiwan that probiotic treatment prevented the transmission of those intergenerational effects. So that was during my PhD, and I got really excited by that. And that's why I kind of started doing these studies in humans. So yeah, I do see probiotics, or, you know, whatever, antibiotics, prebiotics, whatever, things that change the microbiome as a potential target. In terms of being fatalistic, I agree. So that, you know, it doesn't feel great to feel like you're handing down, you know, all of the experiences that you've had during your own childhood. But that's, you know, one of the reasons that we look at these post-adoption, in my case, because a lot of the adversities that we're looking at are foster care followed by adoption, or international adoption after institutional care. So post-adoptive parenting, or more broadly, positive parenting in the context of stress, and how, you know, that variability in parenting can help to moderate the transition of these effects across generations. Yeah.
Great, thanks. Amjad, did you want to ask a quick question?
Yes. Okay, thanks. Great talk, by the way. So I was wondering mostly about the first part of the talk. How would you go about teasing apart the effect of environment and also genetics that also, I imagine, would be correlated between generations?
Yeah.
Just wondering about that.
Yeah, we haven't attempted to do it yet. So that Gusto study is a pretty unusual study for me, in the sense that it's a community sample where we're looking at just kind of within the community experiences of childhood maltreatment. Most of the work that I do is actually, as I mentioned, in children who've been adopted. So that helps to, I guess, remove the genetic effects. And so then we can look at an adoptive parent's experiences of early life adversity, and then how that influences their adoptive children. So that's one way that we're doing that. Within the Gusto study, there's no data that we can access that will help us tease those apart, but hopefully the adoptive work will help.
All right, great questions. Thanks, guys. Michael, do you want to come up and set up? Okay, so Felix has a question. Do you think the effects are purely microbiome based, or could exosomes, epigenetics, vagus nerve properties also play a role? Sorry if I missed this in the presentation.
Thanks, Felix. You did not miss it in the presentation. And yes, I think that they could all have a role. I'm not, as you can see, not very reductionist. I like to include all of the bodily systems. So yeah, I think all of those would potentially have a role. We're looking a little bit at vagus nerve in the lab. There are epigenetic data with that Gusto study, so that's something that we could have a look at for sure. So yeah, I think many different factors at play.
All right, great. Thanks so much, Bridget. So Michael's going to come up and set up. So this is Michael Wells, who's an assistant professor in the Department of Human Genetics in the David Geffen School of Medicine here at UCLA. He completed his PhD in a kind of combination of Duke University and MIT. Is that right? That's super cool. Finished in 2015 and then went on to do a postdoc at Harvard. He's been an assistant professor here at UCLA since 2021, and he's had a lot of awards as well, most recently a K99, and he is a rising star at both MIT, Cornell, and also a host of other institutions from what I could gather. And he's going to talk today about the exploration of human genetic and phenotypic diversity through cell villages.
Hi, everybody. Thank you for joining me today. I'm Michael Wells. I'm originally from Columbus, Ohio, and I've been a PI here since September, I believe, so about 9 months or so. So the lab is pretty new. So my lab uses human stem cell-derived neural models to explore human genetic and phenotypic diversity through a platform we're calling a cell village. Okay, let's go ahead and get started. So our species is characterized by immense diversity in neurological and psychological traits. We think differently, we behave differently, we interact with our environment in different ways. Now, while the origins of these differences have been debated for millennia, we're starting to more fully appreciate that for many of these traits and their associated diseases, there's a certain level of innateness to them, meaning we're kind of just born this way. Now, this has been supported by decades of research, including twin studies that have been able to calculate high heritability scores for diseases like autism and schizophrenia, as well as traits like neuroticism and aggression. In addition, genome-wide association studies and large-scale exome sequencing attempts have been able to identify genomic loci that are actually associated with this trait variance and disease risk. Now, while we're doing a pretty good job of identifying the genetic variants that are showing associations with these different parameters, we're lagging pretty far behind in our ability to understand how these genetic variants influence neurodevelopment. So for example, we don't know how these variants influence, say, how neuroprogenitor cells proliferate and differentiate, or how post-mitotic neurons migrate and form connections with each other. Now, for us to truly make some headway in these efforts, we need to cut across many different levels of biology. We need to understand how these genetic signals influence gene expression in major signaling pathways, and how this in turn affects cellular phenotypes. From there, we need to understand how variation in these phenotypes can actually influence circuit formation and behavioral outputs. Now, as I mentioned, my lab uses human stem cell-derived neural models to investigate these different levels of biology. As a graduate student, I actually focus on the right side of the schematic by looking at mouse models of autism and other type of neurodevelopmental disorders, looking at mostly circuits and behavior. Now, typically, for one to really study these different levels of biology using human stem cell-derived models, if you want to actually look at genetic diversity, your incubator might look something like this. You would have cell lines from dozens to hundreds of different people. You'd put them all into your incubator in separate dishes, and it can look quite chaotic. Now, the problem with this approach is it introduces a considerable amount of well-to-well and run-to-run variation. This can often mask your biological signals. This also introduces a considerable amount of challenges in control and rigor, especially when you're trying to process numerous samples at the same time. Imagine trying to do RNA-seq on 400 samples simultaneously. It's quite difficult. This is also very expensive. and it's very complex, and it requires a lot of people hours, so technicians hate doing this type of work. And overall, this limits the number of hypotheses you can test at any given moment. So as a postdoc in Kevin Eggen's group at Harvard and the Broad Institute, I was part of a team that asked the question, can we do something like this instead? Can we take all those different donors, all those different cell lines, and put them into a single dish? And that's what we did. And that's what we're calling our cell village. So in brief, a cell village is a system in which we take dozens to hundreds of different human donors. We culture them all into the same dish. And then using some bioinformatic pipelines I'm going to describe in here in just a second, we can actually identify relationships among alleles, gene expression, and cellular phenotypes in a high throughput and unbiased fashion. So here's how it actually works. We have our donors. We either whole genome sequence or SNP array profile them, because we're actually going to use that genotype as an intrinsic barcode. So that's how we actually know who is who later on in this process. We use their genome as the barcode. We then convert these cells from these different donors into the cell type of interest. In this case, I'll be talking mostly about neuroprogenitor cells using an induction method that I created as a postdoc that allows us to make NPCs in 2 days from our stem cell starting materials. We then expand, we do our QC. Once we're convinced we have the cell type that we want for all of our donors, we put them all into the same dish. And in doing so, we are actually capturing genetic, molecular, and phenotypic heterogeneity in a shared in vitro environment. So you're probably wondering, okay, now that we put them all together, how do we know who is who? How do we actually extract biological information from a village? Well, as I mentioned, we're actually empowered by 2 bioinformatic pipelines. One is called dropulation, which is based on single cell RNA sequencing of the cells in the village. And one is called census-seq, which is based on low coverage whole genome sequencing. By low coverage, I mean 1x coverage. It's basically about $100 per sample for our census-seq. Let's talk a little bit more about each of these. So essentially, with dropulation, what we're able to do is run single cell RNA sequencing and identify the donor of origin for each sequence cell. We can also, of course, then get gene expression matrices at the donor level. And we can do this because we're actually using transcribed SNPs as the barcode, as I mentioned. We're using natural variation across the donors as our way of reading out who is who. Now, using this, we can actually do things like expression quantitative trait loci detection, because we have our whole genome sequencing information and we have our gene expression information for each donor. In addition, with census sequencing, we can actually extract phenotypic information from our village. Now, the way we do this is we use census-seq, which as the name implies, it's running a census on who's in the village. Okay, so if we say take 100 donors and I do a really good job of counting them accurately, and I put them all into the dish at the same proportion, well, census sequencing should tell me that each donor is 1% of the village. Now, certain things can change that composition. Say time, for example, if you have differential growth rates, or let's say I FACS sort based on the expression of a marker protein. I can then run census sequencing at different times or on different fractions. And in doing so, we can calculate what's called an abundance ratio, which means what is the percentage of each donor in each different fraction or each different census sequencing sample. That actually gives us phenotypic information from our village. And combined, we can actually identify the relationship between phenotypes, gene expression, and genetic variants, and pretty much everything in between. Let me give you a couple of examples really quickly on how we actually use this platform to answer important biological questions. So the first thing we're going to talk about is probably the most complete story. This is work I did as a postdoc, in which we used the village to identify genetic risk factors for Zika virus infectivity. Now, before COVID, Zika was the thing that the news was talking about. This is 2015, 2016. It's basically a virus that's transmitted through mosquitoes that can actually increase the risk of a child being born with severe brain malformations. The most famous of which is microcephaly, or small brain. And in fact, we've since learned that many infants that did not present with any sort of overt birth defect were still at higher risk for developing other disorders, like motor and sensory issues, as well as autism and intellectual disability. So using a village, I performed dropulation on a set of 44 different neuroprogenitor cell lines, so 44 different individuals. And we identified a series of EQTLs. These are genetic variants that influence the expression of nearby genes. Now, one really stood out to me. It was in this gene called IFIT-M3. And in this case, donors that had the CC genotype at this locus had high expression of IFIT-M3, whereas donors with the TT genotype had much, much lower expression of this gene. Now, this is interesting because this gene is believed to play an important role in innate protection from viruses. It's an antiviral protein. And the way it works essentially is it acts as a sentinel system. It can identify endosomes that have RNA viruses in them and shuttle those endosomes to the lysosome for degradation. And so you can imagine that if you have lower expression of this gene, that sentinel system is weaker, and so you increase the chances of infectivity. And this was the hypothesis. The donors with the CC genotype, the ones that have high expression of IFIT-M3, should be more protected from infection than those with the T allele. What's that?
1 minute.
Oh, sorry. I thought you had a question. And then what we did is we used this same village infected with Zika virus and then fact sorted based on expression of the Zika virus envelope protein. We then ran census sequencing on these two fractions, calculated an abundance ratio, and our hypotheses was supported. The donors with the CC genotype had a greater representation in the Zika negative fraction compared to the positive fraction, and vice versa for the TT allele. We then validated this in a non-village setup, meaning each cell line was cultured separately. As you can see, this single variant could explain over a third of the variation in infectivity across our donors. And in fact, when we replicated this in a different set of donor cell lines, we saw this increase to almost 60% of the variation being explained by a single variant in the human genome. Just to close out, we're now moving on into using this system to understand neurodevelopmentals of genetic origin, which is the focus of our lab here. As you can see, we have a growing team of grad students and technicians, as well as my son Misha, who has become a honorary member of the Wells Lab. First, we're using this system to understand 16p deletion, which is and duplication, which is a major cause, one of the most common causes of autism and intellectual disability. We're essentially using a village of neurotypical controls and patient cell lines to identify potential disease mechanisms, as well as phenotypes that are associated with this disorder, including proliferation. We're taking a next step and actually looking under more dynamic conditions, meaning what happens when you have controls and patients, and then you stimulate the system with sonic hedgehog or Wnt, these major patterning factors, these major signaling factors that we know are predominant in developing brain. How do these different cell lines respond differentially to these challenges? And just to close out, we're also interested in expanding the actual phenotypes we can assay in a village to include neuronal morphology, axonal guidance, and hypoxic stress, among others. So the big picture questions we hope to address in our lab have to do with understanding these molecular and cellular mechanisms of these neurodevelopmental disorders, trying to determine if we can find shared biology across different disease genetic states. So looking beyond 16p, do we see some of the same pathways effective in other common causes of autism? And then from there, in the future, we like to translate some of this information to circuit and behavior in animal models. So with that, I'd like to close out and open the floor for questions. Thank you.
Thanks, Michael. That was really interesting. We have time for just one quick question.
Yes.
For GWAS study, from probably say 10,000 to 1 million now.
Yes.
What's the complexity you could do in these cell villages?
So you're asking, so the question has to do with mainly when GWAS studies are done, you're looking at hundreds of thousands of people. We're talking about looking at 50 to 100, exactly. And how are we still able to find things? Very important question. One thing I will say is that what's nice about this system is we're looking at very specific phenotypes in a single cell type, rather than look at these immensely complex traits that have many different factors that are playing a role, genetics and environment. We're able to kind of confine it to very, very simple questions. And in doing so, I think that's when very beneficial to our ability to actually detect individual variants that are causing some of these phenotypes. A very, very important and common question we received. Yes. Yeah.
All right, thank you. We're going to move on to our final talk now. William Zeiger is going to set up here, so I'll just introduce him for everyone. So he is currently an assistant professor in residence in the Department of Neurology, again in the David Geffen School of Medicine here at UCLA. He did his MD PhD at the University of Chicago, finishing in 2013. He did a postdoc at UCLA, which he completed in 2020, and he also got some experience at Hopkins as a neurology resident during this time as well. He also has a number of amazing awards. I really have some amazing colleagues here in the BRI. The most recent of which were the John Lewis Reel Award for Outstanding Research as a Neurology Resident and the Augustus S. Rose Award for Excellence in Teaching. So he really is an all-rounder. He's going to talk today about neural circuit function and dysfunction in stroke and Parkinson's disease. All right, thanks.
Thank you so much. I'm excited to have this opportunity to share with you some of the work that we're doing in our lab. And a lot of our work in the lab is motivated by some of my experiences as a neurology clinician. And in the clinic, we'll frequently find ourselves interacting with a patient who has a new onset of a neurological deficit. And we'll sometimes tell them about the brain's plasticity as a way to draw attention to the fact that the brain may have some mechanisms to promote recovery after an injury. And while we can point to some striking examples of plasticity in the brain, particularly during development, as well as in learning and memory, we don't understand as well how mechanisms of plasticity and circuit function in general are affected in specific adult onset neurological disorders. And I'm going to tell you about two examples of those in the case of stroke and Parkinson's disease. So stroke is the disease we focused on the most to date. Stroke is one of the most common neurological disorders. It's the most common cause of adult onset disability. And in the stroke literature, you'll often run into something called the remapping hypothesis as a way to explain how patients can recover neurological function after stroke. And what the remapping hypothesis suggests is that if you have a stroke in one part of the brain, areas that are spared may remap or change their function to take over the functions lost to the stroke. But there's not a lot of direct evidence showing whether this actually happens or not. So we developed a system to try to study this in more detail, and we took advantage of the mouse whisker somatosensory system. And the mouse whisker somatosensory system has this really nice somatotopic organization in which sensory inputs from an individual whisker are encoded in individual cortical columns or barrels in the somatosensory cortex. And what we can do with this system is we can target a stroke very precisely to one cortical column, and then we can monitor the activity of neurons in the spared barrels in the periinfarct cortex adjacent to the stroke. So here's a little bit of what this sort of experiment looks like. Highlighted here, you can see an infarct that goes through the thickness of the cortex, confined to one very small area of the cortex. We can use macroscopic imaging techniques. Here I'm showing you some intrinsic signal imaging data in which we can map the cortical activity induced by stimulation of a single whisker. So here in magenta, you can see the activity map related to the C1 whisker, and here the adjacent D3 whisker. And in wild type animals, these maps are very stable over months. On the other hand, we can target a stroke just to that C1 cortical activity map, leaving the D3 whisker map intact. We can then use more advanced imaging techniques like what I'm showing you here, 2-photon calcium imaging, to study the activity of populations or even single neurons longitudinally over time. And we can couple that with behavioral assays, like here a whisker discrimination assay that we have set up in the lab. And to make a long story short, what we've found so far is if you look at the primary somatosensory cortex at baseline, and you look at layer 2-3 pyramidal cells, in the C1 barrel, for instance, you'll see that a plurality of neurons respond best to stimulation of the C1 whisker. But in neighboring barrels, you can find some C1 responsive cells as well. That's been well established. After stroke, we ablate the C1 barrel. And what we find is that there are actually fewer cells in surrounding barrels that will respond to the C1 whisker. And those cells that do respond to the C1 whisker stimulation do so with reduced sensory evoked responses. Now, if we let animals just spontaneously recover, we see some recovery in the number of C1 responsive cells over time, but sensory evoked responses remain depressed. We also put animals through a forced use therapy paradigm to sort of mimic a rehabilitation technique that's done in patients with strokes. And what we find there is that sensory evoked responses become potentiated, but we never see sort of true circuit remapping where you would have cells changing their function to take over the functions that were lost to stroke. And so we think this means we may need to engage additional plasticity mechanisms in order to sort of achieve true circuit remapping and improve recovery after injury like stroke. And so now we're trying to get at those additional pathways to promote plasticity. And in particular, we're interested in parvalbumin interneurons as potential regulators of post-stroke plasticity. And so we're using transgenic animals that allow us to specifically label parvalbumin cells in vivo. So here you can see parvalbumin cells in the primary somatosensory cortex labeled with fluorescent protein, and we can track individual cells for months after a stroke. And so far, we've found that the number of PV cells in the periinfarct cortex seems pretty stable after stroke. We're also using similar methods, but expressing genetically encoded calcium indicators to look at the function of these cells over time after stroke. So here's some preliminary data looking in the normal healthy brain, where we are examining the spatial distribution of whisker responses in PV cells. We're also looking at the function and changes in responsivity over time in response to plasticity-inducing paradigms. And recently, we've completed a study looking at how changes in whisker responsivity occur in PV cells over time after stroke. And we have collected all that data, but we're in the process of analyzing it. So switching gears now, in the clinic, I primarily see patients with Parkinson's disease. And we often think of Parkinson's disease as a movement disorder causing problems like tremor, stiffness, slowness, balance problems. But many patients with Parkinson's also have cognitive impairments. And if you take a population of patients with Parkinson's disease and follow them for 10 or 20 years, in fact, the majority of patients will go on to develop dementia and problems like hallucinations. And it's thought that there's actually two patterns of cognitive impairment involved in Parkinson's disease. The first are what we call sort of frontal and executive impairments. And these seem to be related to loss of dopaminergic activity, primarily in frontal circuits. And they don't predict well who will go on to develop dementia. On the other hand, there are what have been termed posterior cortical impairments, problems with visual spatial function, visual perception, hallucinations. And these are related to the deposition of a protein called alpha-synuclein in Lewy bodies in neocortical circuits. And importantly, this pattern of posterior cortical impairments more strongly predicts progression to dementia. Furthermore, if you look at the brains of patients with Parkinson's disease, those that have these posterior cortical cognitive impairments also show selective hypometabolism and atrophy in posterior cortical structures. And so this suggests that these posterior cortical circuits may be particularly important for the development and progression of cognitive impairment to dementia. And so we're interested in studying this in more detail, and particularly asking a very basic question, which is how does alpha-synuclein pathology within individual neurons actually affect circuit function in vivo? And so what we're doing is we're using some fluorescent tools, in this case a dye, which allows us to stain aggregated alpha-synuclein within neurons in vivo. So this is some ex vivo data just showing specificity of this dye for aggregated alpha-synuclein. And then this is some in vivo data showing that we can actually label these aggregated alpha-synuclein aggregates within neurons in vivo. We can then couple this again with genetically encoded calcium indicators and follow changes in the activity of individual neurons over time as pathology develops in these animals. And we're working to couple this with behavioral assays in which we can measure visual perceptive function in our mice using the same types of tests that are impaired in patients with Parkinson's and dementia. We're also interested in the reciprocal question, which is how do disruptions in circuit function affect the progression of alpha-synuclein pathology? So there's accumulating evidence that in Parkinson's disease and other neurodegenerative disorders, the misfolded or aggregated protein-based pathology can spread throughout different circuits within the brain over time. But we don't really understand the factors that govern that spread. And so we are interested in understanding how alpha-synuclein spreads from subcortical structures like the striatum, which are involved in movement disorders, to the cortical regions, which are involved in cognitive impairment. And so we're setting up systems to use chemogenetics to specifically modulate the activity of circuits within the brain to see how changes in activity in these specific circuits drive progression of alpha-synuclein pathology to cortical circuits, where they then presumably participate in the progression to dementia. So with that, I want to acknowledge the work of everybody who's contributed to this, in particular, our lab manager, Brenda Vasquez, and a postdoc in our lab, Baruch Campos, who have been doing all the work, imaging PV cells in vivo. Of course, my mentors, particularly Carlos Portera, who was my postdoc mentor. We have a group of exciting collaborators that we're working with as well, and always interested in more collaborations if you find any of this interesting. So thank you very much.
Okay, thanks.